Today we are discussing about circulatory system, part 4. Clotting of blood or coagulation. In this picture, you can see blood clot. We can study in detail. Steps involved in clotting. Do you know what is a clot or how it happens? Yes, if there is any injury or any wound happens in your body, within few seconds you can see clot. Then there are different steps are there in clotting. First step, injured tissue cells and platelets releases thrombokinase. What is thrombokinase? Thrombokinase is an enzyme produced by platelets. At the time of injury, platelets releases an enzyme. It is called thrombokinase. This thrombokinase, what happens to this thrombokinase? Thrombokinase is helping to activate prothrombin into thrombin. Activate prothrombin into thrombin with the help of calcium ions. Then calcium ions, where it is present in the plasma. Then prothrombin is also a substance present in plasma. Prothrombin will convert into thrombin with the help of thrombokinase and calcium ions. All of you understood? Then the next step, this thrombin in presence of calcium ions, again with the help of calcium ions, thrombin will convert fibrinogen into fibrin. What is fibrinogen? Fibrinogen is a protein. All of you know that we already learned in circulatory system part 2, right? This fibrinogen into fibrin with the help of calcium ions. Who will convert fibrinogen into fibrin? Thrombin will convert fibrinogen into fibrin. This, where it is present, fibrinogen is also present in plasma. That is a protein present in plasma. Then with the help of calcium ions, thrombin convert fibrinogen into fibrin. Fibrin, what is fibrin? Fibrin is an insoluble protein. Insoluble protein present in plasma. Then the microscopic threads of fibrin are sticky and form a network at the wound. Then fibrin form a network around the wound. Then that is a microscopic thread. Fibrin is a microscopic thread. It is an insoluble form. Insoluble protein present in plasma, that is fibrin. Then the next step, the blood cells are trapped in the network of fibrin. We know that this fibrin is a microscopic thread, insoluble microscopic thread present in plasma. This microscopic thread network of fibrin will trap blood cells. Then the network then shrinks and squeezes out the rest of the plasma, which is in the form of clear liquid that is called serum. Yes, here what happens actually, blood cells are trapped. At the time of injury, within few seconds you can see clot. Yes, then how it happens? These blood cells, this fibrin is an insoluble form of protein. This fibrin network or microscopic network of fibrin trap the blood cells. Then these blood cells with the fibrin, that we call it a solid mass. That solid mass left behind is called clot. That means what is clot? Clot is a solid mass left behind is clot. Then how it happens? with this fibrin trap the blood cells. Then blood cells plus this insoluble fibrin altogether form a clot. Then what is left out? The network then shrinks and squeezes out the rest of the plasma. You can see if you have any injury happens, within few seconds clot will occur. Then a white colorless liquid will come out. This colorless clear liquid we call it as serum. Then serum means you already learned that in earlier chapters, circulatory system 2. What is serum? Serum means the plasma from which protein fibrinogen removed. That means the activated form of fibrinogen is fibrin. This fibrin with the blood cells that is removed. The remaining part is serum. Then blood cells and plus fibrin, that we call it as clot. The technical term of clot is thrombus. Very important in examination. The technical term of clot, that is thrombus. No platelets, no coagulation of blood. Viral dengue fever causes abnormal decrease in platelets, leads to hemorrhage. You know that, yes, platelets count will be very less if there is, as we got dengue fever. Then it leads to hemorrhage. The next, fate of blood in clotting. What is the fate of blood at the time of clotting? What happens to blood? That we are discussing. Here we already learned that what are the steps involved in clotting. Then here what happens that time? Here you know that blood consists of two main constituents, corpuscles and plasma. Then plasma is a liquid part. Corpuscles means cells present in blood. Then in this corpuscles and fibrin. Fibrin is a protein, is an insoluble protein present in plasma. This fibrin is present in plasma, that fibrin plus this corpuscles. Fibrin combined with corpuscles and form clot. The remaining part of plasma, that means without fibrin, that is called serum. Do you understand? Right? We can discuss about some other topics: blood transfusion, blood groups, ABO and RH systems, compatibility and incompatibility. Compatibility and incompatibility of blood. Blood transfusion. What is blood transfusion? Blood taken from a healthy person is introduced through one of the patient's veins. That is called blood transfusion. Then donor will donate blood to recipient. Recipient means who is receiving blood. Donor means who is donating blood. Then donor will give blood to recipient. Then that is called blood transfusion. But one condition is there to donate blood, that should be compatible. That should be matched with recipient. The donor's blood should be matched with recipient's blood. Then if that is not matching with the donor's blood, the recipient blood is not matching with donor's blood, that we call it as incompatibility, or or incompatible blood. If that is matching, both donors and recipients blood is matching, then we can say that is compatible blood. ABO blood group system. ABO blood group system you can see in human beings. Mainly 4 types of blood group you can see in human beings. Blood group A, blood group B, blood group AB, blood group O. Then based on the type of antigen present in the blood group we are classified into 4 based on the type of antigen. What is antigen? Antigen is a protein present on the surface of RBC. Then based on the type of that particular antigen, we are classified blood groups. Example, antigen A. Antigen A is present in blood group A. Then in blood group B, which antigen is present? Antigen B. In blood group AB, that contain which antigen is present? Antigen A and antigen B. But in all blood group, there is no antigen. There is no antigen on their surface of RBC. Then another is important point you have to remember. Based on the antigen, complementary antibodies are produced in the plasma of that particular blood. Plasma of that particular blood, complementary antibodies are produced. Yes, here you can see blood group A contain antigen A, but antibody, complementary antibodies are produced. That antibody we call it as anti-B antibodies. Anti-B antibodies are produced. In blood group B, which antibody is produced? Anti-A antibodies. In blood group AB, there is no antibodies in blood group AB. But in blood group O, anti-A antibodies and anti-B antibodies are produced. But there is no antigen in their blood. Then in blood group O, 2 types of antibodies are there, anti-A and anti-B. But in blood group AB, 2 types of antigens are there. That is antigen A and antigen B. Blood group AB has no antibodies. Blood group O has no antigens. Next is incompatibility and compatibility of ABO blood group. You can see here which blood group is matching and which blood group is not matching. The matching blood group we call it as compatibility. Mismatching blood group, incompatible blood. Then here you can see is O blood group receiving blood from only from O blood group. It's mismatching to blood group A, B and AB. But it is only matching with blood group O. Blood group A is matching with, or yes, blood group A can receive blood from blood group A and blood group O. Then blood group O can donate blood to blood group A, then blood group A. A and O can donate blood to blood group A. Then blood group B can receive blood from where? It can only receive blood from blood group A, O and blood group B. This mismatching with blood group A and blood group AB. But blood group AB can receive blood from all the type of blood group. All the type of blood group it can receive blood. Then But O can donate blood to all the type of blood. O can donate blood to all the type of blood. That's why O we call it as universal donor. Then AB we call it as universal recipient. AB is universal recipient because it can receive blood from all the type of blood. We can discuss ABO blood group, the main points. Universal donor. Which is a universal donor? Which blood group? Blood group O. Universal recipient? Blood group AB. The concept of blood grouping was discovered by Karl Landsteiner. The concept of blood grouping was discovered by Karl Landsteiner. Then the surface of RBCs have special proteins called, we already discussed, that is an antigen, called an antigen. Antigen is a special protein present on the surface of RBCs. Then you have to know that in human beings mainly two types of antigens we can see, antigen A and antigen B. Based on the type of antigen, the blood groups are classified into 4. Then based on the antigen in its that particular person's blood, complementary antibodies are produced. Then where it is located? It is located in the plasma of the blood. Plasma of the blood. Then antigen A has which type of antibody? Yes, antigen A is present in a blood group A. Then that type of blood group, which type of antibody you can see? You can see anti-B antibody, right? Anti-B antibody you can see in blood group A because it contains antigen A. RH system. It is a type of blood grouping. The blood of most people contains a substance called RH factor. Then that the presence of RH factor in their blood, that we call it as RH positive blood. The person lacking RH factor in his blood, then that we can say RH negative blood. In this diagram you can see a special protein present on the surface of RBC, that is called RH factor. Then that blood we call it as RH positive blood. Then this RH factor is lacking in some blood, then that we call it as RH negative blood. The factor was first discovered in rhesus monkeys. It is first discovered in rhesus monkeys. That's why the term came RH factor. RH positive person donate blood. Suppose an RH positive person donate blood to an RH negative person, then the blood of recipient, RH negative person, that is the recipient, RH negative person develops an antibody for RH substance within about 2 weeks of transfusion. Within 2 weeks, an antibody will produced in RH negative person for this RH substance. Then if at second transfusion be given to RH negative person, second time if that person is getting that recipient, that means RH negative person is getting RH positive blood, the antibody produced in first transfusion causes a reaction with transfused blood. Then it will lead to death. That it will causes a reaction with transfused blood because that recipient is getting again RH positive blood. Then there should be a reaction and it may lead to death. RH factor in pregnancy. Yes, When an Rh negative mother carries Rh positive child, the negative mother carries positive child, the first child will be normal. The second child, if conceived soon, may have a problem. It lead to the death of fetus. Then the mother is Rh negative, but the child is Rh positive. There is an antibody produced in the plasma of mother's blood against the Rh factor, because the first child has is Rh positive. Against that, mother's body will develop an antibody against that Rh factor. It's already developed. Then the second child is conceived soon. Then that is also a positive child. Then this antibody will fight against, or the antibody will react against child's blood, and it will lead to death of fetus. Revision. Exam based questions and answers we can discuss. Write the location of the following. Antigen A on the surface of RBC of a person having blood group A. Anti-A antibody in the plasma of a blood of a person having blood group B. Rh factor on the surface of RBC. Fibrinogen. Where is it present? Fibrinogen is present in the plasma of blood. Prothrombin is also present in the plasma of blood. Name the following. First one, the insoluble protein present in the plasma of blood. Insoluble protein present in the plasma of blood, which is that fibrin. The iron helps for coagulation of blood. Which ion? Calcium ion. The enzyme released from platelets during injury, that is thrombokinase. The technical term of clot, yes, that is thrombus. The cells initiates the clotting of blood. The cells initiates the clotting of blood. Platelets. The antibodies present in the plasma of blood group O. Anti-A antibody and anti-B antibody. Antigen present in the RBC of blood group B. Which antigen? Antigen B. Write the functions of the following. Thrombokinase. Thrombokinase, with the presence of calcium ions, it will convert prothrombin into thrombin. Platelets. Next is platelets. What is the function? Platelets helps to initiate the process of blood clotting. Initiates. The term you have to use. Thrombin. Thrombin, with the presence of calcium ions, it will convert fibrinogen into fibrin. Then calcium ions. Calcium ions helps for blood clotting. You can explain how it will combine with thrombokinase. Thrombokinase, with the presence of calcium ions, yes, it will convert prothrombin into thrombin. With the presence of calcium ions, thrombin will convert fibrinogen into fibrin. That also you can write. You can explain in that way. Define the following. Blood transfusion. What is blood transfusion? Transfer of blood from donor to recipient. That is called blood transfusion. Then thrombus. What is thrombus? Thrombus means blood vessels are trapped in the network of fibrin and form a mass. This is a solid mass that we call it as clot. The technical term of clot is thrombus. Then that solid mass we call it as thrombus. Thank you, children. Have a good day. Like, share and subscribe.